Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore gene control in eukaryotes. At the end of this lesson, you will be able to state that transcription factors are proteins that bind to the DNA and are involved in the control of gene expression in eukaryotes by decreasing or increasing the rate of transcription, and explain how gibberellin activates gene by causing the breakdown of DELA protein repressor, which normally inhibits factors that promote transcriptions. Let's get started. Transcription is the process of copying genetic information from DNA into mRNA. It occurs in all living cells, but it is more complex in eukaryotes compared to the prokaryotes. Two major differences account for these increased complexities are multiple RNA polymerase. For example, RNA polymerase 1, 2, and 3 are involved in the transcription. However, in prokaryotic cells, they have one type of RNA polymerase as compared to eukaryotic cells with 3 RNA polymerase. And there is a requirement for additional protein. So in eukaryotes, RNA polymerase cannot bind directly to the promoter. They must interact with transcription factor and other type of protein to initiate transcription. So RNA polymerase 2 transcribe protein coding gene to produce mRNA. So research by Robert Rodel in 1979 shows that RNA polymerase 2 require additional protein to begin transcription in eukaryotic cells. So this demonstrated the need for initiation factor in eukaryotic cells transcription. So these essential proteins are known as transcription factor, or commonly known as TF. So they are sequence-specific DNA binding protein that regulates the rate of transcription by binding to the particular DNA sequence near the target gene. So in this diagram, you can see that this is a promoter region. The green color is the gene. So we have different or multiple transcription factor, at least must have one. They bind to their recognition site, which allow RNA polymerase to bind and initiate the transcription. And these transcription factors are important because some of these transcription factors are produced based on or in response to the environmental or stimuli. So what are the functions of transcription factor? So they serve as general transcription factor, so which they are required for all transcription by RNA polymerase 2, and they form part of the transcription initiation complex at the promoter, which results in the initiations of transcription. They regulate gene expression by switch on and switch off at the correct time, in the correct cell types, and at the correct expressions level. So basically, in eukaryotic cells, we do not have operator, but we use transcription factor to help us to switch on or switch off the gene expression at a specific time. So imagine that if you want to switch on a particular gene, we produce transcription factor. But when we want to switch off a particular gene, we simply degrade particular transcription factor. And some of these transcription factors act as activator to increase the likelihood of RNA polymerase to bind to the DNA and initiate the transcription, or some of them act as a repressor by blocking the binding of RNA polymerase to the DNA. So basically, transcription factor can be a positive regulator or a negative regulator. And transcription factor can involve in the developmental control to activate the gene in a specific sequence to ensure correct body pattern formation. So basically, we need to know the timing, the correct timing, so that we will be able to develop a particular cells and make the cells become a specialized cells for particular organ. Next, okay, transcription factor also involved in the sex determination, where the control or this transcription factor control the expression of gene that determine sex in mammal, whether male or female. And this transcription factor can respond to the environmental changes by activate stress response gene in reactions to environmental changes. So they can be or they can regulate the cell cycle as well, where the product of the proto-oncogene and tumor suppressor gene act as a transcription factor to control the cell division or mitosis, growth, as well as apoptosis. Apoptosis is a way, or we call it as the programmed cell death. And as well as dose function, transcription factor actually may have hormone, or it contribute to the hormone actions, because hormone influence the transcription by activating or inhibiting specific transcription factor. Some hormone bind to a protein or a receptor to serve as transcription factors. Let us look at the role of DELA protein in gibberellin signaling and seed germination. So gibberellin is a type of plant growth regulator, or commonly known as a plant hormone, that control key developmental transition, including the change from the seed dormancy to germination. So in barley seed germination, gibberellin acid or gibberellin release from the embryo stimulates the production of alpha amylase in the ailerons layer. And this enzyme hydrolyzes starch stored in the endosperm into glucose, which the embryo absorbs for growth. So the idea here is, if the environmental condition is not suitable for the seed to germinate, so therefore the seed will continue to be in the seed dormancy. However, during seed germination, where the environmental factor is conducive, is optimum for the seed germination, then the embryo releases this gibberellic acid in response to the environmental change. For example, when there is a sufficient water supply. And due to the production of gibberellin acid or gibberellin, a series of reactions take place, result in the production of alpha amylase, and alpha amylase hydrolyzes starch stored in the endosperm to form maltose and subsequently form glucose, and glucose is used as the respiratory substrate for the embryo to grow and to carry out respiration first. So at molecular level, there is a protein known as DELA protein, and this DELA protein acts as negative regulators of gibberellic acid or gibberellin signaling by preventing target gene transcription. So you can see that in this diagram, DELA protein bind to the transcription factor and prevent the transcription factor to bind to the promoter. So without transcription factor to bind to the promoter, therefore RNA polymerase can't bind to the promoter and initiate the transcription. And GID1 in this diagram is the gibberellin receptor. So in the absence of gibberellic acid or gibberellin, DELA protein bind to the transcription factor, and the name of this transcription factor is known as PIF, or phytochrome interacting protein. And the binding of DELA protein to transcription factor PIF, preventing promoter binding and blocking transcriptions of target gene. So there is no production of alpha amylase. Therefore, there is no germination. However, when gibberellic acid is present due to the favorable germination condition, for example, adequate water, the embryo now releases gibberellic acid into surrounding tissues. So gibberellic acid now binds to its receptor GID1, causing a conformational change, and therefore the activated GID1 bind to the DELA protein, forming GID1 gibberellic acid DELA complex. And this is very important because the subsequent reaction helps to break down DELA protein through an event known as polyubiquitination. So polyubiquitinated DELA is degraded by the proteasome. Proteasome basically something like proteases. And the removal of the DELA protein allows PIF to bind to the target's promoter and therefore activate the transcription of the alpha amylase gene and results in the germination. So we can see that in this diagram. So for our syllabus, a very easier way to describe it, gibberellic acid bind to the receptor, and this result in the breakdowns of the DELA protein in a series of reactions to free the transcription factor PIF. So the PIF now bind to the promoter, RNA polymerase bind to the promoter and initiate the transcription process where alpha amylase gene is transcribed to produce alpha amylase. And alpha amylase break down starch to form maltose, and maltase break down maltose to form glucose, and this glucose can serve as respiratory substrate for the cells to carry out respiration. And therefore, with the production of ATP, the cells manage to undergo anabolic reactions and germinations take place. So, and with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.